Hello everyone, I'm Koyan and today we are going to learn how to answer questions related to chapter 11 which is related to immunity in humans. It's quite a short chapter, but most importantly you have to know how to differentiate between first, second, third line of defense, what's going on with that, and the types of immunity, diffusion active immunity, passive immunity. Yeah, those are the important parts of this chapter. So it's quite a short chapter. But before I would like to enter, I would like to show you in general form what did we actually learn in first, second, and third line of defense. I know there are a lot of mechanism we learn, but what group is it involved in? Okay, so the first line of defense is the very general one. It's basically the one where they needed to act as a barrier to prevent the pathogen to enter. So the examples are right here: skin, lysozyme, mucous membrane, hydrochloric in stomach, and blood clotting mechanism. Because blood clotting, they not only want to prevent from severe blood loss, they also want to prevent, when the blood clot quickly, it also will prevent the pathogen from entering your skin. And yeah, skin is true, maybe shedding on the skin, because our skin, we will have old dead skin, it will also shed off to prevent, pathogen from sticking onto it. And yeah, lysozyme is basically the enzyme that will destroy the pathogen. So they can be found in tears, mucus, saliva, and mucous membrane, which lysozyme and mucous membrane is quite related lah, because mucous membrane, which lies in the respiratory tract and digestive tract. Yeah, there are mucous membrane. They will keep secreting mucus, and one of the function of the mucus is to act as a barrier to prevent pathogen from entering. And yeah, hydrochloric acid in stomach, okay, it will kill the pathogen, those stuff. And the second line of defense is whereby we learn fever, phagocytosis, and inflammation. Then for fever, we actually learn what's the point of having fever. Yeah, it can actually increase the phagocytic activity and promote phagocytosis. And phagocytosis, you have to know what's happening in there, okay, which later on we'll mention. And inflammation, we also have to know more about what's the point of the tissue releasing histamine when it is for injured tissue, why do they release histamine? What's the function? And last but not least, you know, when when the third line of defense is when they manage to go through the first and second line, when the pathogen succeed on entering, now it enters the third line of defense. So this would actually stimulate the immune system to produce antibodies to destroy all the pathogens. So antibodies is more specific already. So yeah, they are classified. For our lymphocytes, there are T lymphocytes and B lymphocytes. Okay, so T lymphocytes is basically the one that will kill the infected cell, and it is also the one that stimulate B lymphocyte to produce memory cell. Meanwhile, what does B lymphocyte do? B lymphocyte is the one that produce antibodies, and it is the one that produce memory cell. But it needs to get stimulated by his friend, which is T lymphocytes. And T lymphocyte, they straight out will kill the infected cells lah. Okay, but B lymphocyte, they will produce the antibody to destroy the infected cell. So now let's take a look at the question right here, which is Selangor Set 2, 2023. Yeah, it shows a second line of defense mechanism, which is basically the phagocytosis that involve leukocyte. So in specific, all right, I know leukocyte, they classified into many types. Okay, so for leukocyte, they actually have granulocyte, a granulocyte, and if you want to look into detail, right, a granulocyte and granulocyte. What's the difference between granulocyte and a granulocyte? A granulocyte means it does not have the grains. Okay, but this one it got granule, not grain, sorry, granule. Alright, it doesn't have granules in it. So this leukocyte is basically white blood cell. So they got differentiate into two types, a granulocyte and granulocyte. And a granulocyte, it consists of um, lymphocyte and monocyte. Meanwhile, for the granulocyte, it consists of NEB. What are those NEB? Is neutrophil, eosinophil, and basophil. So this monocyte, right, is actually still immature. It will become mature to be called as macrophage. Okay, so yeah, these are the leukocyte we actually learned in the chapter 10, which is very in detail what are their functions. But most importantly for this chapter, we will more involved in lymphocyte, monocyte, and neutrophil. But normally we won't mention like monocyte, neutrophil, or macrophage. We will just mention it as phagocyte because monocyte and these two are actually phagocyte. Or you can say macrophage lah, to be precise, because macrophage is the matured, I mean activated one. So these two are actually phagocyte. So what do phagocyte do? Phagocyte actually does phagocytosis. So it's the one that engulf the pathogen. Meanwhile, for lymphocytes, they are more specific, which is involved in your third line of defense. Yeah, so got B lymphocyte, T lymphocyte. That's how you differentiate between them. So let's take a look at this one. They are talking about phagocytosis. So it's basically involving phagocytes. So who are the phagocytes? It's either macrophage or the neutrophil lah. But they didn't mention in precise, so it's fine. Okay. Because maybe because SPM. So yeah. For this phagocytosis, a lot of students don't know the arrangement of it, don't know how to really understand like what's going on with it. So it's actually quite similar. The starting of, I mean, it's actually quite similar to how the amoeba species obtain its nutrient. It also requires a pseudopodium to extend and then engulf the pathogen, and also requires the lysozyme, the enzyme, to break down this uh, bacterium. So how do you actually understand the whole process? Is basically what happens at first is this pseudopodium. It will extend and then yeah, it will just envelop the bacteria. So yeah, P is a pseudopodium, and then here got bacterial lysozyme. So lysozyme is actually a vesicle in the cell. And then yeah, later it will fuse with it and then it will secrete something called as lysozyme, which is an enzyme, digestive enzyme. So first of all, the pseudopodium, the the phagocyte will actually extends its pseudopodium, and then it will straight out will envelope the bacteria. And then it will actually, through this ingestion of bacteria, it will actually form a phagosome. So Q is actually a phagosome. Okay, this is due to the pseudopodium envelop the bacteria, so it ingests it. So bacteria ingestion, it form a phagosome. And after you form the phagosome, the lysosome. So lysosome, don't mix up with lysosome and lysozyme. So lysosome is the vesicle containing the lysozyme, and yeah, it will basically fuse with the phagosome together. So it will lysosome combine with the phagosome. Lysosome combines with the phagosome. And what does the lysosome do? Lysosome is actually the one that will secretes the lysozyme. So secretes lysozyme into the phagosome. So you can see that, yeah, it secretes the lysozyme enzyme into phagosome because they are already combined together already. So after they secrete this enzyme, what does the enzyme do? Then you further elaborate that this enzyme will destroy the bacteria. Destroy bacteria. So after you get to destroy the bacteria, can you see this part? They already being broken down. So the bacteria, some of it will be absorbed into the cytoplasm, while some of it will just straight out expel lah, because it's unwanted material, undigested material. So yeah, the waste product will get expelled. It will get expelled out of the cell. Alright, so these are the whole process of what happening in this phagocytosis process, which is, yeah, a lot of info already. So the most important thing is when they saw a bacteria, no matter what type, yeah, they straight out just extend pseudopodium and envelope. Okay, extend and envelope it. And this bacterial ingestion will eventually form this thing called a phagosome, which is Q. And later on, the lysosome right here, it will combine with the phagosome. So this lysosome, it will secrete this lysozyme enzyme into the phagosome, secretes lysozyme, and yeah, this enzyme will basically destroy the bacteria. So after you destroy bacteria, the nutrients you get absorbed into the cytoplasm. Meanwhile, what happens to the waste product? They will expel out the waste product. So most important keywords is extend pseudopodium, envelope, and then bacteria to form this phagosome. Then lysosome combines with phagosome, and this lysosome secrete the lysozyme. Lysozyme destroy the bacteria and expel the waste product out of the cell. So P and Q, basically what I've mentioned already. P is pseudopodium and Q is actually the phagosome. So what about the role of substance found in lysosome? What is the substance found? The substance found is the lysozyme. So it basically is a digestive enzyme. So you can say that it digests, or you want to write it in another word, it destroys the bacteria. Moving on to this process carried out by leukocyte in the body defense mechanism. So it's straight out this very obvious phagocytosis process, which is carried out by phagocyte. So yeah, and make sure you spell it correctly, phagocytosis. So for the inflammation, yeah, the damaged tissue will actually release a histamine, which stimulates inflammatory response, leads to function of the histamine. So if you can actually recall it, let's take a look at this part. Yeah. So you know, when there's an injury, the damaged tissue will actually release the histamine. So what does the histamine do? It can actually cause more blood flow to, yeah, flow around there. Because why do they need more blood flow at there? It's basically to accumulate the clotting factors to carry out the blood clotting mechanism. And how do you actually let the red blood cell, flow and accumulate at there, the injury part area? Since they are always contained inside the blood vessel, right? So this histamine is the one that will increase the permeability of blood capillaries against the phagocyte cells. So yeah, they will basically allows the phagocytes, the red blood cell, to flow out from the blood capillaries easily to actually accumulate at the injury area. Yeah. So first of all, it will actually allow the blood capillary to expand. Okay. Allows blood capillaries to expand. So after it allows blood capillaries, okay, initially it like that. So it allows blood capillaries to expand. And this blood capillary, they actually have pores in the blood capillary. So they got basically some small hole, but we actually didn't really learn that. So this blood capillary will expand and then it will actually allows yeah, the red blood cell, the phagocyte, to actually squeeze out from the blood capillaries more easily. So what does this mean? It actually means in another term we have to mention in the more specific way, which is increase the permeability of blood capillary to the phagocytes, so that the phagocytes could actually flow out from the blood capillary to accumulate at the injury area, because the injury area contains those pathogens from external environment. So they want to carry out phagocytosis at there, to quickly ingest all the pathogens to prevent those bacteria pathogens from further entering your body lah. So they quickly, want to initiate this inflammatory response. Yeah. So that the main reason is basically to allow the clotting factors and phagocytes to accumulate. Clotting to accumulate in the injury area, infected area, because there got a lot of the pathogens. So why does the clotting factors have to accumulate there? Is basically to do the blood clotting mechanism. Yeah. To quickly, form a scab to prevent more pathogens from entering your skin. And then for phagocyte is basically they want to do the phagocytosis as quickly as possible. That's the reason why they quickly escape out from the blood capillaries. All right, so moving on to the next one. So these two are the functions of histamine. Now let's take a look at this part. So this is basically a very specific action, because you can see the shape has to be complementary. So it emphasizes the specificity. Okay, so it's basically it involves antibody and antigen. So it's the third line of defense. So label antibody and antigen. Yeah, a lot of students get messed up between antibody and antigen. So antibody is actually the one that shaped like Y. It looks like Y shaped one. Yeah. So these are actually the antibodies. And antigen is actually present on the cell surface membrane of the cell. So this antibody, yeah, it will basically bind to it and then eventually breaks it lah. So this is basically the antibody and this is the antigen. So antigen is the one that will stimulate your lymphocytes to produce antibody. So your lymphocytes produce all this antibody to quickly bind to the specific antigen and then yeah, destroy the pathogen lah. So state the name of one leukocyte that produces antibody. So it's very obvious that it is the lymphocyte. Lymphocyte. If you want to be specific, you can mention B lymphocytes also can. Yeah, because to be specific, B lymphocyte is the one that produces antibodies, but the answer scheme just straight out allows you to just write lymphocytes lah, or else a lot of students will lose mark on this part if they didn't write B lymphocyte. So yeah, like what I mentioned just now right here, we got lymphocyte, monocyte, neutrophil, eosinophil, basophil. Remember that monocyte or the macrophage lah, and the neutrophil are the phagocyte that involve in phagocytosis, which is in second line of defense. What about third line of defense? Are the lymphocytes that produce antibodies. Yeah, okay. And then they want you to define what is actually antigen. So it's actually the protein that can be found on where? On the surface membrane of the cell. So is protein that antigens are protein that I know the grammar got a bit cacat, found on the cell surface membrane on the surface of the cell. It is basically, yeah, it will actually stimulate the immune system, immune response, because it is the foreign particles that enters the body. So antigens are foreign particles. You take at, yeah, take a look at the textbook. Yeah, it is a foreign particles that enter body and stimulate the immune response. That enter body and stimulate immune response because they will be identified as something foreign. So they will actually, beep or beep or stimulate the immune response to produce antibodies and find it out. Lah. So yeah, they recognize it as a foreign particles known as antigen. So yeah, it's the one that enter body and stimulate the immune response. So this is actually the meaning of antigen. So you can actually just point out this one, you can actually get 1 mark already, or you just based on what you learn from textbook is actually the foreign particles that enter body and stimulate immune response. Yeah, you get 1 mark for that. But actually, the reason why I stuck halfway because I suddenly thought of something that I learned before. I mean, not in SPM lah. Is antigen? It can actually there are actually self antigen and non-self antigen. So the antigen we are learning is non-self antigen, which is the foreign particles that enter body. They have a non-self antigen, but our own body cell, we also have our own antigen, honestly lah. But yeah, it actually won't stimulate the immune response, or else it will therefore happen this autoimmune disease lah. But yeah, don't learn too much in detail later. You complicate yourself. Just learn in SPM you do. Okay, so the next question right here. A student was infected by the pathogen, and then how explain the action of antibodies towards antigen in this situation. So how are you going to explain based on the diagram? So a lot of students don't really read the question properly. They are mentioning based on diagram, but maybe students just straight out explain action of antibodies, just write out agglutination, precipitation, uh, neutralization. They just straight out mention all the action of antibodies they have memorized. But they want you to base on diagram. So yeah, remember to read the question properly. You must to base on this diagram. So right here, they basically bind to it and then they will break it, right? And that you can see lysis occur. They broke it in the end. So how are you going to explain it? First of all, antigen found on surface of pathogen. Okay, a lot of students get confused. Whether should I write pathogen, bacteria, virus? Wow, so many types. Which one should I actually write? So pathogen is the general term for all of them. So if you are not sure whether it's a virus, is it a microorganism, bacteria, you just straight out write pathogen is the most safest way to answer it. Unless the question got mentioned, oh, it's a bacteria, then you just write bacteria lah. Like just now, this one is actually a bacteria. So yeah, it is a bacteria. Is they really direct to you this is a bacteria. But if you are unsure whether is it pathogen, bacteria, yeah, you just write it's. I mean, you are unsure is it a bacteria, virus, microorganism, all those stuff. You just straight out write the most general term, which is pathogen. Okay, but phagocytosis normally is for the bacteria one. They destroy the bacteria. Okay, but for this one, you're not sure, ma. They didn't mention is it, bacteria? Is it a virus? So you can straight out write it is found on the surface of pathogen. So the antigen that is found on surface of the pathogen is identified as something foreign. So it will do what? Like the police, they see bad guy, okay, something foreign. It will stimulate immune response, people. So it will stimulate immune response or the immune system lah. So the immune system to produce or the lymphocytes to produce antibodies. So it will stimulate the because we want to straight out mention yeah, it will actually stimulate the antibodies. So the production of antibodies. So what do antibody do? Stay there and do fashion show? No lah. So this antibody, it will bind to it, right? But this one, they just straight out bind lah. They didn't show any more complicated diagram. So yeah, it will actually bind to this antigen. They are complementary towards each other. So since they are specific lah, so you have to mention it will bind to it. Antibodies, bio favorite word is called bind. So it binds with antigen. So you must write out this specific word right here to get marks lah. Sometimes they are very strict with it. So it binds with the antigen. So after that, what does it do? It binds to it and do what? It broken down already, right? So yeah, the diagram actually help you a lot. It's like Bina ayat, you see the action and then you write out the complete sentence lah. So causes bacteria to break down. But this is in a bio way, so a lot of students find it difficult to break down. Or you can say lysis. Lysis. Lysis is also the meaning of breakdown occur. So the main point is it is found on the surface of pathogen, which will stimulate the production of antibodies. So these antibodies binds with it, causing it to break down. So alamak, this one I write pathogen. Yeah, you just write pathogen all the way lah, to not confuse the person who marks it. So yeah, you can just, this is how you get 1 mark right here. This is based on the answer scheme lah. So there are more than 3 marks lah. So either 3 of them, yeah, you get 3 marks. I mean, any 3 points, sorry. So now let's take a look at this immunity in humans. You can see this person crying, okay, basically showing you this is the tears. What does the tears contain? It actually contains the lysozyme enzyme. Right? So explain the mechanism in 9.1 to avoid pathogens from entering the body. So this is, yeah, you can see it's very simple diagram. But a lot of students panic, don't know how to explain. It's actually think it as a very simple way because this is just SPM. So just try to mention what you can see. Oh, it secrete the tears, right? So what is this line of defense? It is actually the first line of defense. So just mention what you have learned. This is the first line of defense. So by secreting the tears. So what does the tears contain? It contains lysozyme, or you want to mention antimicrobial protein, which is what written based on the textbook. So what does the lysozyme do? It will destroy the pathogen. Right, so you just mentioned this is the first line of defense. Okay, by secreting tears, the tears contain lysozyme. Then this lysozyme, it will destroy pathogen. That's how easy it is. Yeah, so 0.2, 0.3, 0.4. So any of these 2 points, you actually get 2 marks right here. Yeah, the explanation is actually quite direct lah. Yeah. Now let's take a look at this. This is actually the phagocytosis. So let me revise with you again. This is the pseudopodium. Pseudopodium saw this bacteria, so it will extend. So after extends, it will envelop the bacterium. Eventually, the bacteria ingestion, it will form the phagosome. So after it formed the phagosome, the lysozyme, lysosome will combine with the phagosome. So this lysosome, it will secrete lysozyme. Sorry for the confusion. It will secrete lysozyme. So what does the lysozyme do? It will break down, or you can say digest the bacteria. So after you digest the bacteria, what do? Yeah, you actually absorb the nutrients, and then what about the waste? It will get expelled out of the cell. So these are the 6 most important steps you have to know in the phagocytosis process because they normally want you to like explain lah. And this one they talk about, yeah, he stepped on a glass while cleaning kitchen, causing infection, effects of the body defense mechanism that act on the bacteria. So explain the effects, the body defense mechanism. It is basically wanting you to explain the phagocytosis. Okay, so the way they ask is a bit weird, like explain the effects. Okay, normally we will just ask, explain the body defense mechanism that act on the bacteria we do lah. Okay, effects is kind of weird lah. So yeah, these are the points you can actually mention, which what I've already mentioned with you. just now, just that they got add on something lah. And the most general thing, like you really don't know what is this whole process in detail, but you know what is the main thing. It is a phagocytosis. Remember to write, it is a phagocytosis process that occurring lah. And yeah, the detail part, if you remember, is very good because you can score majority of the marks already. Now let's take a look at this part right here is two methods on how baby obtain immunity. So this is basically breastfeeding. And then by looking at this one is the immunization schedule. So compare the immunities obtained by the baby through these two methods is actually very direct. The comparison is actually very frequent type of question that we ask in SPM. So you really have to understand it and memorize it. So most importantly, you must to understand it or else very hard for you to memorize the points to write onto it. So the immunity, this one I'm not going to mention a lot. It's just basically facts right here. So the similarities, you can write few of them and then differences. Because they want you to compare immunity, so don't forget you have to not only talking about differences, you also have to mention the similarity because they will allocate marks like maybe list 1 marks for similarities to get the full marks. Yeah. So how do you actually see that? This is actually the vaccine. Vaccine. So vaccine is the one that containing dead weakened pathogen. So when you inject this vaccine, those dead weakened pathogen, it has the antigen on its the pathogen surface now. So it will stimulate the immune system to produce antibodies to fight for it. So while producing the antibodies, they will also produce the memory cells. So next time during the infection, the memory cells that are produced, they can quickly identify this pathogen. So they can straight out produce antibodies in a shorter amount of time and more antibodies, more concentration of antibodies to straight out fight for it. So this one, it is the one that produce antibodies by its own. So when it produce antibodies by its own, it's called an active immunity. But how can you see is it artificial or natural? It is very obvious. All this vaccine, do you think it comes in a natural format? It grows from the tree, no, right? Okay, it's from someone, no, it's not in natural way. We synthesize it from the lab, so it is artificial. So it is an artificial active immunity. So active immunity means you produce antibody on your own. But let's take a look at this breastfeeding right here. So yeah, actually the mother's milk contains a lot of antibodies from the mother. But now the baby basically drinking, sucking the uh mother's milk. So breast milk lah. So this breast milk actually contains antibodies from the mother. So the antibodies from the mother enter the baby. So the baby, it doesn't produce antibodies from its own I mean, based on this breastfeeding situation, you can see it just straight out obtain antibodies from external source. So when it obtains from an external source, when you obtain antibody, obtain, I write AB on that, from external source. So who is this external source? This external source is actually from the mother antibody. So it is a passive immunity. So how can you see is it artificial or natural? So this is basically the most natural way. It's not artificial, okay? Okay, so it comes in natural form. Okay, so it's a natural passive immunity. So when you want to compare, you not only have to say passive or active, you also have to measure is it natural or artificial. So the direct one, yeah, just write out passive, natural, active, artificial, active, yeah. And then you can emphasize the antibodies comes from where? It is produced by the mother. And then for method 2, since it's a vaccine, it is produced by the body itself. So the vaccine doesn't contain antibody. The vaccine contains the weakened pathogen. So this weakened pathogen is the one containing, how to say, so yeah, you go inject this vaccine. So this vaccine right here, it contains pathogen. I mean, it's a weakened one lah. It won't cause disease. So it's a that or weakened pathogen. This is a vaccine. So this pathogen, like just now, it contains these foreign particles, which is the antigen from this pathogen. So this will actually stimulate the immune response of the body. So your body will produce the antibody to fight it off. So the antibody, it will fight off the weakened pathogen, which is from the vaccine lah. So this vaccine doesn't contain antibody. Okay, the antibody, they detect this weakened pathogen, which is this vaccine content. Then your body will produce antibodies to fight it off. So at the same time, it will produce memory cells. That's the reason why it is actually long lasting. Yeah, the effect is long lasting. And yeah, there's actually a lot of them. So you can actually go through the information, but yeah, that's the reason why it is long lasting, because memory cells are produced. But if you rely from the other people, other external source of the antibodies, of course, you didn't produce antibodies on your own. So at the same time, memory cells are not produced. That's why the immunity is just temporary. And the others, you can just take a look at it. It's actually quite easy to understand. And then, how are you going to explain this inflammatory response when he stepped on a nail that contains bacteria and then cause it to become inflamed? So it is basically mentioning about this inflammation now. So how are you going to explain this inflammatory response that can destroy and neutralize harmful organisms, microorganisms, and toxins at the early stages of infection? So this one is basically talking about the histamine. Okay, the histamine, okay, the damaged tissue, it will produce this histamine. So what does the histamine do? Like what I mentioned previously, the question got asked. It will actually stimulate an immediate inflammatory response. So it will cause your blood capillaries to expand more, and yeah, for more blood flow to the infected area. And also, yeah, it will increase the permeability of the blood capillary to the phagocytes. Okay, and the phagocytes can easily escape out from the blood capillaries to flow to the infected area and accumulate right there, so that blood clotting mechanism is triggered and phagocytosis can occur. So this is the straight out whole point of it. It will release, stimulate immediate inflammatory response, expand more blood to flow the infected area, increase permeability of blood capillaries to phagocytes, and then it will accumulate. Then, since the clotting factor accumulate at the infected area, blood clotting mechanism is triggered. What about phagocytes? It will carry out phagocytosis right there. And then they want you to compare immunities obtained by the individual through these two methods. Oh, they want you to state the name of substance X and Y first. So it is injected with substance X to give him immunity against tuberculosis disease in the future. And this one is straight out after bitten by a snake. So because snake got this thing called a venom, yeah. So yeah, it will is actually quite dangerous lah. And you have to straight out go to get inject with this thing called antiserum. because our body, right, in order to produce antibodies, it takes quite a long time. But this one, the venom already enter your body. You want to quickly solve it in a short period of time, or else this individual will straight out mati, die. Okay, so you have to quickly obtain, the fastest way is to obtain antibodies from an external source. So when you obtain external antibodies, you got more antibodies, ma. Yeah, so yeah, actually fight it off lah. So it is actually a passive immunity since you obtain antibody from an external source. And yeah, it's injected with a substance called antiserum. It is a artificial way. Okay, and for this one is the vaccine, because in the future, it's not like when he kena tuberculosis, then he only inject, right? Like COVID-19, you can actually relate to it a lot lah. So probably because of this pandemic, we can understand it better lah. Okay, so this is actually a vaccine. So the substance. So how are you going to mention? You cannot just straight out write vaccine and antiserum. The marker, the examiner don't know are you mentioning X or Y. So you have to say substance X is vaccine, substance Y is antiserum. They only want you to mention the name. Okay, I know some students they memorize like, oh, I know the whole thing about it, so they straight out right. Oh, the vaccine contains blah 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 blah. But they just want the name. So substance X is vaccine, substance Y is antiserum, compare the immunities. So this one is actually active immunity and is also artificial. Okay, so this comparison right here, both protect body from infectious disease involving antibodies. Just that one is external source, one is it produced by its own self, and both are artificial immunity. So you draw it out in a table for differences. Can see straight out right here. What does the vaccine contain? You can compare it. Yeah. And then this is actually for prevention. Let's say in the future, you may also expose to the tuberculosis also. And this is straight out immediate protection. Or maybe you going to a place where you have a high risk, okay, of getting bitten by the snake, maybe, then you can also get it also. But um, the thing is, it is actually not last long lah. It won't last long, so it's temporary. Since it's the antibodies from external source only. So in the end, the antibodies will also die lah from the external source. And then it gives immediate means it straight out protection, quick, very quick. But this production of antibodies by your own body, right, it actually takes a long process lah, honestly. And the reason why it takes a long process, actually, we got learned in detail if you further get to learn biology in detail. It's basically because like you need phagocytosis process, and then you need a lot of activation and stimulation because B lymphocyte, T lymphocyte, they need someone to actually stimulate it. And yeah, then only will in the end produce antibodies. So it takes a longer time lah. So but the good thing is it will produce memory cells. So this immunity will last for a longer period of time. Then yeah, this one is before being infected. But this one you can be before and after being infected. Before because yeah, maybe you go to a place where it's highly exposed to it, to that risk. And yeah, these are the comparison. Okay. Now this question, Negeri Sembilan one, is talking about blood body mechanism to stop bleeding. So it is actually not really talking about the inflammation. It is the one that talk about stop the bleeding. Compared to just now the question, they said, oh, neutralize the toxins. No, where is the question? destroy and neutralize harmful microorganisms and toxins. So we are mentioning about inflammatory response. But this one, they say stop the bleeding. So it's actually what you learn in chapter 10, blood clotting mechanism. So yeah, for that, I will actually explain it in the maybe another video if I got time. So because yeah, it seems kind of crazy because they got a lot of enzyme involving in it also. But basically, these are the process that occur right here. And yeah, you when your when there's a wound lah, the platelets will actually clump at the wound. And then what happens next is the clotting factors will actually stimulates the platelets to form thrombokinase. So thrombokinase is an enzyme. Okay, thrombokinase with the help of vitamin K and Ca2 plus, it will actually convert prothrombin to thrombin. Thrombokinase will convert prothrombin to thrombin. So with the help of this vitamin K and Ca2 plus, that's why vitamin K, this vitamin is actually very important for blood clotting. And then this thrombin right here is also acting as an enzyme to convert fibrinogen to fibrin. Okay, this one last already, okay? No more straight out another process. Okay, this fibrin, finally lah, it is the one that will form network on the blood, like spider net like that lah, okay? So it will form a network on the wound to prevent the red blood cell from escaping lah. So eventually it will track the red blood cell to form a scab, okay, and then eventually it will close the wound. So when you close the wound, you will eventually stop the bleeding. So this is how everything answers it all lah. So first of all, platelets come at the wound, and then yeah, the clotting factors will actually form this thrombokinase. So this thrombokinase, with the help of vitamin K, Ca2 plus, will convert prothrombin to thrombin. And this thrombin will actually convert this fibrinogen to fibrin. This fibrin is the one that will form the network, okay, on the wound. Sorry, why did I write blood? Okay, on the wound, and it will actually trap the red blood cell to form scab. So eventually it will close the wound. Alright, and then when we are going to explain the injured part, the finger becomes swollen, red, painful, so you can see because of the inflammation lah. So explain the body defense mechanism against bacteria. So it also talking about the histamine. So yeah, you just straight out the same thing lah. Okay. So you have to really understand the histamine function. Okay, basically want the phagocyte and the clotting factor to accumulate at the wounded area or the infected area. So how do they actually let them accumulate? It's actually causing the blood capillary to expand for more blood to flow and increase the permeability of the phagocytes so that they can straight out, yeah, squeeze out from the blood capillaries and go to the infected area so that, yeah, blood clotting and phagocytosis can occur. Okay, then these questions right here is the injection of weakened suspension of pathogen. So this is a pathogen. So it is a vaccine. What about this? It contains specific antibodies. So it is a antiserum. If you still can't identify, okay, this one straight out getting the antibodies from external source. So when you straight out getting antibodies from external source, it is the passive immunity because your antibody won't produce by its own now. You just straight out rely from the external source. But this one right here is a weakened suspension of pathogen. So they inject this pathogen to stimulate your immune system to produce antibodies. So you got produce antibodies by your own. So this is a active immunity. But you have to specific like is it artificial or natural lah? Then how are you going to state the difference? So yeah, it's actually very direct right here. Okay. Yeah, it's the same thing lah. I'm not going to mention too much. Yeah, all right. And then moving on to this last question is talking about measures to prevent the spread of AIDS. So they just show you the HIV. So you just mentioned the measures to prevent. So we actually learn like how HIV is spreaded. Like, I mean, how HIV is transmitted is actually sexually transmitted disease (STD). So other than through the seminal fluid or vaginal fluid, it can also be transmitted through blood. That's why those, people that take drugs, some of them they will share needles, right? Yeah, it can also be transmitted through the blood also lah. So yeah, the measures are actually a lot of them. Okay. Alamak. Yeah, but a lot of students they don't understand like why this HIV is so hard to be cured compared, if you talk about I mean cure lah, like or you treat it. Normally they can only treat the symptoms but can't really cure it lah. So the reason why it's so hard to destroy this virus is because they will mutate easily lah. They mutate very frequently, so it's very hard for them to, find out a way to destroy them because their structure always mutate. So yeah, in the end it's quite difficult lah. And then other than that is because it eventually straight out just kind of hijack your lymphocytes lah. So the reason why they want to target the lymphocytes instead of like other cell, maybe your muscle cell or other cells lah, red blood cell, all those things. The reason why they should target lymphocyte is because lymphocytes they actually actively divide a lot, especially when there's an infection occur. Lymphocytes are the one that is actively dividing most of the time because we are constantly exposed to pathogens. So lymphocytes are always actively dividing. So they target lymphocytes because they will straight out will, use their genetic material to to combine, to kind of like manipulate lah, okay, the genetic material of our normal host cell so that our own host cell will actually, since they already modify the genetic material with their genetic material, yeah, eventually our own cell will produce the HIV virus lah. Okay, so there will be more HIV virus to be produced from our own cell because our own cell, the DNA is being hijacked lah, okay. Because this HIV, they are very smart, okay. They will use their DNA to insert into our host cell DNA right there. So our host cell, we just straight out, code what's in the DNA ma. So we just code out the protein. So yeah, they will basically code out this more new virus, which is this HIV virus. So yeah, you actually lead to a destruction of the immune system. But in detail, I won't explain it that much lah. So how are you going to preventive measure? Okay, so avoid unprotected sex with individuals who may be infected. So the way to do it is through maybe using condom, and don't share contaminated needles, because right now we are trying to avoid transmission through the blood. So we can say about, avoid sharing of needles, and then do not receive blood from contaminated HIV virus. But normally they will check lah your blood whether it has this virus, and followed by this one. Not only not only those two like previously, it can also be transmitted from mother to the children. Okay, and yeah, like from through the placenta also can, if the mother is the AIDS patient. Yeah, the mother can also transmit it to the child lah. So they will actually take this antivirus during pregnancy and delivery. Yeah, this one is actually more detailed lah, and try to avoid breastfeeding. And last but not least, normally just conduct this awareness campaign lah. So it's actually very BM, because in Bahasa Melayu, a lot of kebat question, right? You actually mentioned about awareness. Okay. So yeah, that's all for this chapter, and I will see you in the next chapter. So thank you so much for watching. Thank you so much for your time, and bye bye.